Okay, so as you know, today's sort of focus is immunogenicity, and I'm going to be speaking to you a little bit about a number of different tools that are available. I'm going to be focusing on ProImmune's tools and technologies. Some of you will have heard some elements of this before, but there's also some new data as well that I'm going to be sharing, which is quite exciting, just came out very recently. We've had some fantastic introductions already from speakers, obviously this morning with Amy, but also yesterday as well. So I'm going to try not to obviously just recapitulate a lot of the same things, but at the same time, rather than just show the same old slides, what I want to do is to try and give us some insights, particularly into some of the assays and the kind of sorts of data that really these can help you achieve and the kind of the questions that they can address. So what I'm going to do is focus on, as I mentioned in my sort of introduction slides yesterday, the elements of the strategies that we have really focus on the immune response in a very broad way. So thinking about antigen processing, presentation, generation of T and B cell responses, and innate immunity. And to do that, I'm going to focus on a number of the different technologies, including the antigen presentation assay ProPresent, which some of you will be more familiar with the sort of acronym MAPS, which is MHC-associated peptide proteomics. Muta, which is going to come at the end of my talk, which is a kind of a new technology involved in protein engineering and actually how that can help address some of the big challenges with immunogenicity particularly. I'm going to talk about T-cell proliferation assays and dendritic cell T-cell assays and the parts that they can play in both risk management approaches, in development of different biotherapeutics, as well as pinpointing some very specific areas of maybe your immunotherapy, monoclonal antibody, for example, that is maybe driving some unwanted immune responses. And then also I'm going to talk about cytokine release assays, so talking a little bit more to innate immune responses and cytokine storm. So this introduction slide is something which I think is important just to sort of bear in mind, that immunogenicity is not something that's very easy to understand. It's not something that one assay or one strategy is going to come up with all the answers to. That's really very clear from many years of experience. And I think also it's very clear to understand from also what Amy was saying that the biology of your own disease that you're studying and investigating is really understood as well. So immunogenicity to biological molecules is a complex area. Most of us in the room here are thinking about the medical interventions, so particularly for tumour therapies, for infectious disease, for autoimmune disease. But we have to also think about the design of any protein, whether that's for medicinal use or maybe other situations where food proteins, for example, are also being manipulated. Then, of course, the immune response can also be generated against that particular protein. And unwanted immunogenicity is a big problem, we've heard that, and can result in treatment failure. And there are a complex set of intrinsic and extrinsic factors which come into play. So we're talking about the actual extrinsic elements, so the route of administration, how a protein is delivered, how often it's being delivered to an individual, the dose, the patient's immune system status, of course, whether it's hypo or hyper-responsive, whether there's any pre-existing ADAs, whether they're neutralizing or not, and also the HLA restriction of those individuals. These are all a broad set of different factors which come into play and can be very difficult to dissect out what's contributing different elements to the unwanted immune response. Intrinsic elements such as the amino acid sequence itself, how a product is formulated, what the aggregation status of that product is, and maybe a little bit more intrinsic factors that we can sometimes have control over and sometimes not. And it is a very complex area. So we know that managing immunogenicity risk is really key to the success of a product. If you have a product which is going to market, which is going to have high immunogenicity, you might as well not bother, because it's absolutely imperative, because that means that not only the patient safety is considered, and but also the potency of the drug as well. Now, no cartoon of an immune response can ever do it justice, but this is really very simplified, of course. And what we're talking here is that a therapeutic or a protein of any kind is taken up by an antigen-presenting cell, and these cells are then presenting peptides in the context of MHC molecule, in this case MHC class 2, because the protein is exogenous. And these peptides are typically, on average, about 15 amino acids in length, but will vary from anything between 12 to 20, and sometimes outside that remit as well. And this MHC peptide complex presents to the T-cell receptor on the surface of the CD4 helper T-cell. And this cognate interaction, along with the appropriate co-stimulation, leads to T-cell activation, T-cell proliferation, and production of the appropriate factors such as cytokines, which will then go on to help the naive B cells become memory B cells, and plasma cells, which are then going to be producing antibodies. Now, of course, these antibodies are really what we're trying to prevent, the binding to the target, because that in itself can result in loss of efficacy, and in serious cases, as in the heart situation with the MAGE-A3 cross-reactivity, you can actually even have death. So clearly, we want to prevent all of these things happening. So a lot of the assays I'm going to be talking about use a number of different cells, and the ProImmune PBMC biobank is a kind of a key component of the different assays that we provide. The first thing is that the donors are sourced from the UK population in general, which is where we're based, and our labs are in Oxford. And that's quite nice because we actually have a broad HLA distribution in general terms, and that means that we can select donors of a broad broad HLA distribution, but also we can specifically, if required, look at specific HLA types if that's of interest. So some diseases are associated with different HLA types, or there's maybe a known predisposition to presentation on a particular allele, and that is something that we can also help you with. So our donors are fully HLA typed using third-gen sequencing, so we get full resolution of the full class 2 type, DR, DP, and DQ. And those donors are then used in a number of different assays. Many of the times we'll be working with monocyte-derived dendritic cells, which is a kind of a culture-based approach, but we're also using PBMCs as well from these biobanks. So the first assay you've heard about already, the MAPS assay, and I'm going to be talking more focusing on MHC class 2 today. The great thing about this technology from its early days, where there was a kind of limited success in sensitivity, we've now really been able to marry the beauty of proteomics and the high sensitivity of proteomics with immunology to get to the bottom of some of these really key issues that have been dogging immunologists for a very long time. So it's great that we've now been able to really get to the heart of the matter. And it allows us to identify naturally processed and presented peptides from any protein or mixture of proteins. So the beauty is that we can look at either a single therapeutic protein, we can look at a complex mixture of proteins if that's also required, and actually see what is being presented on the surface of the antigen-presenting cell. We can use it to address different questions. So are there different elements of your protein that are being presented, such as a neo-epitope from a junctional region that's been introduced as part of the design strategy of your program? Maybe we want to understand what the impact of protein modifications is on antigen presentation. That's also a key question. And of course, the approach can also be used on the flip side of thinking about targets for vaccines or other sort of immune therapies. We can look at MHC class 1 or class 2, and in this talk I'm really going to be focusing on class 2. Emily already talked about class 1 yesterday. And from the start of this process, the whole set of data is typically delivered in around 4 to 6 weeks, so it's very rapid. We have a whole set of different publications, and I'm going to take you through some case studies just to give you an example of that. So the principle is that the MHC class 2 pathway means that we can actually start with protein provided to us by the clients that we're working with. We typically need around about 20 milligrams of protein, so that's something to take into consideration, but that's not an absolute number. We can work with you if that's a bit of a high target. And that's the sort of the amount that's required if we're doing a 10-donor study. We would then generate monocyte-derived dendritic cells, and these DCs are immature at that point, and they're very happy to take up antigen. So we can load that with your proteins of interest. The DCs are then matured, and they upregulate MHC class 2, and they obviously naturally process the peptides from those proteins and present them in the context of class 2. We then carry out an immunoprecipitation step, so that the cells are lysed. The antibody that's specific for either DR, DP, or DQ is used to pull down those MHC peptide complexes. These complexes are then eluted, the peptides are eluted from the groove, and these are then sequenced through tandem LC-MS-MS. And what we do is we generate a database, which is the whole human proteome, and on top of that we add in the protein therapeutic sequence, or the mixture of proteins that we're working with, or that can be a whole proteome. For example, if this is an organism, like a bacterial organism, and working with a bacterial lysate that's been loaded, then of course you can just add that in. And what we report are all the peptides from the loaded material that are presented. Now, in antigen presentation, of course, there are thousands and thousands of peptides presented, and what we're reporting are all the peptides from your protein, not all the peptides in total, because what you need to do is to specifically search for the peptides that can be from your original protein. And it's a very, very sensitive method. We're able to look at essentially femtogram quantities of peptides. And the beauty of it is, of course, that you can look at overlapping peptides from the protein of interest. And the number of peptides that are identified, which is a common question, is very much driven by a number of different factors. So the assay has been optimized to work with a wide variety of different protein types. But if you're working with a large protein like factor VIII, then of course you will proportionately identify more peptides than if you're working with a peptide-based therapeutic that's a single 20mer, for example. It's just a matter of how the cell's obviously going to be processing that individual peptide or that individual protein. We can look at DR, DP, and DQ. DR certainly is more prevalent in regards to the total number of peptides that you'll identify, and that's what most people choose to look at in the first instance. But of course, there are many situations where DQ and DP are also very important, and ProImmune is the only provider that can look at those alleles as well, those allele families, those loci. And that's really, really important to know because it gives you the bigger picture. You can really understand exactly what's going on across the board. So this is a case study that was from a paper that was published just a few weeks ago. It was a group that was working in Singapore and in Europe, Danone, you may have heard of them if you've been eating yogurts recently, a big international company with food ingredients. Now, one of their products is infant milk formula. And one of the big challenges at the moment, or has been for a while, is really that cow's milk protein is the leading cause of food allergy. And that really is driven by the fact that you just don't get oral tolerance generated in those individuals. And this can be a big issue, particularly in infants up to 3 years old just on infant formula. And the most typical way of treating that is actually to use hydrolyzed cow's milk-based infant formulas. This is often used for individuals who are at a higher risk of developing an allergic disease. And that means basically that the formula itself, if you look at the milk protein, the intact protein is full length. If you do a partial hydrolysis, then you get shorter fragments of the protein. And then and this is like a prevention formula, but in mild to moderate allergy, there's an extensive hydrolysis that is performed on the protein. Whereas in severe allergy, really the only option to you is to give the component amino acids rather than the whole protein in any form at all. Now, the key thing as well is that obviously we're thinking about just peptide fragments that might be inducing an unwanted immune response. But actually, the sort of flip side is true that presentation of T-cell epitopes is also a prerequisite to inducing that tolerance. So identification of peptides that are actually being presented from this milk protein is really important in the generation of Tregs. And that's something which has been known for quite a while. So this particular project is where Danone came to us. They wanted to understand particularly beta-lactoglobulin, which is a very key component. It's the most abundant protein in whey and one of these major allergens in cow's milk formula. Now, T-cell epitopes from beta-lactoglobulin have previously been shown in animal models to reduce allergic skin responses. And so this particular portion of the protein, position 13 to 48, has been well characterized. But what they wanted to do is to look at hydrolyzed milk formula protein to actually look and see what which peptides from that protein were being presented. So we performed ProPresent on a panel of 12 HLA-type donors. And this is focusing on HLA-DR in the first instance here. And what we were able to do is to identify peptides from beta-lactoglobulin as part of the project. So the donors, the HLA types are listed here. We have a broad range of different HLA restrictions, including some that are more sort of common to more common to Asian allele families, so DRBR405, 0404 as well. So it's important to have a breadth of different HLA types. This data slide here just shows you the repertoire of peptides that were identified from this one protein. We have the individual donors listed on the y-axis here in the table, and then the individual fragments that are identified are shown as well. And what you can see for MHC class 2 are these overlapping peptides, which is quite typical. So you have an overlapping region. And so in this particular donor here, the P2 donor, 2 individual peptide fragments were identified from that protein, literally just overlapping and shifted along by 1 amino acid. So for every identified peptide, we report an expect value, which is a bit like a p-value in short term, so it gives you a confidence of identification of that particular peptide sequence. So an expect value of less than 0.05 is indicative of peptide identity. So you can be confident that that peptide is present. If you don't see a peptide present in ProPresent and in any other sort of mass spec assay, it doesn't mean to say that peptide isn't there. That's one thing that's really important. No assay is perfect. So if a peptide is not identified, then it may be that the peptide just hasn't flown well in the mass spec. It may be that the abundance is below the limit of detection of the assay. But if the peptide is identified, then you know it is there. And the great thing about this as well is that we can actually look at post-translational modifications to a certain degree, such as phosphorylation, oxidation. A whole range of different PTMs can be looked at in this assay as well. So if you know that and you want to specifically look for those mods in advance, then we can also help you out. The exception, and one of the, again, common questions, is glycosylation, and that is a massive challenge because obviously the complexity of sugar chains and the different options that are available does mean that that's not an option that can really be looked at in the simple way through this particular assay. So in short, I mean, this just sort of summarizes obviously all the data. We've actually identified 2 unique sequence groups. So these are the different donors out of the 12 that we're identifying peptides. So not all donors, you will always identify peptides from the protein that's been loaded. So that's one thing why it's important to look at a breadth of different donor types. And you can see here that donor 2, 3, and 12, we're identifying this particular region, and then donors 4 and 7 were identifying this other region as well. And these, importantly, both of these peptides that were identified overlapped with the previously identified region of interest from the beta-lactoglobulin. They then performed themselves a functional T-cell proliferation assay to confirm those peptides were functional, and this also demonstrated that the beta-lactoglobulin-derived peptides, obviously we showed they were presented by HLA-DR when incubated following the hydrolysis. The other case study I was going to talk about was introduced by Zubin yesterday. We already heard a little bit about the Treptocog alpha story, the engineered factor 7A. Now, we know that factor 7A has been used for many years with no problems, but when Novo Nordisk were working with this protein, they wanted to generate a fast-acting, increasing the enzymatic activity of the protein. And so they introduced 3 amino acid substitutions. So still very good homology, but nevertheless, 3 substitutions compared to the native sequence. Now, this only became apparent in the phase 3 trial, but with a retrospective analysis, which was, I think, really very much the purpose of the paper, goes to show that if you do your homework at the very beginning, some of these big challenges and expensive problems, not to mention the fact that the patients were affected, could have been foreseen at the very beginning. So once the anti-drug antibodies, or these inhibitors, were measured, of course, the trial had to halt immediately. And the question was asked, well, how can we make sure that these neoepitopes are not engineered in, leading to the development of inhibitors? So this is just one part of the study, looking at the ProPresent, the MAPS assay, which was one element of the paper. And the nice thing was, this Novo Nordisk came to us to do this study, provided us with the factor 7A, and completely independently, they also performed this assay in-house. So the great thing about this is basically we were able, these are the 3 different amino acid substitutions in the protein, and we were able to identify exactly the same regions coming out, both compared to Novo Nordisk, where they did a 17-donor study, and the ProImmune study with 12 donors. So these are different donors. The assays aren't aligned in relation to the protocols, but it is a very robust assay, and I think this is demonstrated by the fact that the same core sequences were identified in both independent studies. And furthermore, when you go on to then look at the functional relevance of those peptides in a T-cell proliferation assay, then if you look at the wild-type sequences shown here in blue, this N-terminal modification at position 158 actually had no impact at all on T-cell proliferation. So this is a peptide that's presented, but actually doesn't lead to any T-cell proliferation. So that's another important take-home point, that not all peptides that are presented are necessarily functional T-cell epitopes. That's very important to bear in mind. However, the C-terminal mutations that are quite close to each other, position 296 and 298, at those two particular positions, you can see that the impact of introducing that change has led to a strong T-cell response. And then when you look at combining those 2 different peptides together, again, that's demonstrated and confirmed. So the conclusions from the paper, through the T-cell proliferation assays, the antigen presentation assays, and also physical MHC peptide binding assays, was that immunogenicity was the root cause failure of this drug. Out of the 3 mutations, 2 out of 3 of them were presented by HLA-DR, and they bound with a high affinity to those DR alleles. And unfortunately, these early stage trials were just too small to detect some of these potential immunogenicity issues. So actually, if they'd done this kind of analysis in advance, although there wouldn't have been kind of an assurance that that was a problem, at least that would have been flagged up, as something to drill into in more detail. So it is important to sort of show and understand that these kinds of preclinical assessments, although they can't prove what's going to happen in the clinic, that's why we do clinical trials, they can certainly alert us to any specific challenges that might come out. So I mentioned already that DP and DQ is something that's very important, and again, Zubin kindly sort of presented this in a slightly different way yesterday. This is data from CSL Behring, where they looked at comparing full-length factor VIII, Afstilla, which is their B-domain deleted factor VIII molecule, and also plasma-derived factor VIII, all in the presence of von Willebrand factor. And this abstract was published last year. And what you can see here is when you, the key here is the three different colors. So we've got DR peptide presentation in blue, DP in sort of orange, and then DQ in red color. And the three different versions are on the left here. So we've got the full-length factor VIII, the Afstilla B-domain deleted, which is why there's no peptides being presented from the B-domain, and the plasma-derived factor VIII as well. And the key visual thing that you can see is that every little part of these dots is a different fragment of peptide that's presented. So for factor VIII, you get a whole set of peptides being presented across different alleles and different loci, DR, DP, and DQ, with DR, as we would expect, being the most prevalent. For Afstilla, in the presence of von Willebrand factor, without obviously the fact there are any B-domain peptides present, so the overall presentation has to be lower, we can actually see that despite that, there is actually a significant reduction in the presentation of DR peptides particularly, and also the other loci as well in these other domains. And that's compared, also when you look at the plasma-derived factor VIII as well, this is obviously clearly reduced compared to the full-length factor VIII. So there's something about the recombinant generation of these proteins, the factor VIII proteins, which is actually altering the antigen presentation. And that's something which people are still trying to understand and investigate, and we need to understand more about maybe protein refolding, protein expression, and actually what the effects are on antigen presentation. So once you know that your peptide is presented, then one of the questions is, well, yes, is it a functional T-cell epitope? Is this actually something that we need to be worried about? Now, of course, you could go straight to these sorts of T-cell proliferations from the very beginning. You can do overlapping peptides for the whole region of your protein. And that's something which can be done, of course. And the challenge with that is really the numbers of peptides that we're talking about. Sometimes the numbers of cells are limiting for each donor that we're running in these types of assays. And so it's quite nice in some circumstances to be able to narrow those down using the antigen presentation assay first to know that those peptides are actually being processed and presented. The other point where you can use these sorts of assays are over specific engineered regions, whether that's a potential neoepitope that's been introduced at a junctional region, whether that's specifically over the CDRs of a monoclonal antibody. Again, there are many, many different circumstances when you might want to run these particular types of assays. We work with healthy donors, again, typically look at panels of 40 or 50 donors, but we're flexible with the number, so we can look at 20. We typically wouldn't recommend anything lower than 20 from the point of view of significance of the data, but most of the time you want to get a good cohort of different donors representing a broad tissue type distribution. And if we're doing an overlapping strategy, then we would generate 15 peptides overlapping by 12 amino acids, high purity, so 95% purity, or alternatively, we can actually use those peptides that have been identified using ProPresent, and that's another nice option. Going back to the post-translational modification point that I made earlier, if there were any specific modifications on those peptides that had been identified in ProPresent, or you knew in your own product had a modification at that particular position, then of course these synthetic peptides can also be generated with those modifications and then tested in these assays. It's a simple 7-day culture with the peptides. And what we do is we take, it's a flow cytometry based assay, which gives you great flexibility at addressing deeper questions if required as well. It's a very sensitive assay, and it means that we stain the PBMCs that have been depleted of CD8s. We stain the PBMCs with CFSE, which is a fluorescent intracellular dye, and they're incubated with the peptides with no exogenous cytokines or factors. And then over those 7 days, the T cells proliferate if there are cognate interactions occurring, and the cells then divide, and the CFSE is then diluted every time there's a division of the cell. And we look at the 7-day period because that's when most of the divisions have all occurred. And of course, the nice thing is we're gating on live lymphocytes. we're gating on CD4 positive cells. There's no ambiguity about what the type of cells are. And if needed, if you're interested, we can also do additional gating and phenotyping of the cells to look at and drill down at other population requirements if needed. So what you do is you basically get a map of the overall peptides. Each of these different colored bars is a donor that's responding significantly to the peptide. So what you can see is like a hotspot area here where your peptides are responding to a larger number of donors. So in this particular instance, in the CDR3 domain, this is actually from adalimumab. You can actually see the number of donors responding to that portion of the antibody is higher than in the other domains as well. And actually, this is interesting when you do tie it in with the antigen presentation assay. You can actually see that there are typically quite common peptides that are either in the CDR domain or sometimes in the framework region, which very, very commonly come up in the antigen presentation assay. And there's a lot of, it's very, very interesting when you see these types of peptides that are being presented. Just a few nods, because everybody knows what these peptides are. We kind of see them a lot. And they're not exactly the same sequence, but they're all a very, very similar family. And that's really just these framework peptides are very, very commonly presented. And it's very interesting to see how those all perform as well in the T-cell assays. Maybe you just don't care about the individual epitopes yet. Maybe you're just at the very early stage where you've got proteins and you just want to know, have I got a problem or not? Do I need to care about this? So I would always caution that approach to a certain degree. I mean, sometimes that just has to, that's a situation we find ourselves in. But I think it's also good to have a risk-based strategy where, depending on your molecule that you're working on, you might really want to do some of these different assays in parallel rather than just think one at a time. A lot of these can all be done in parallel, and that's an important element just to consider. However, if you do want to do a comparative protein antigenicity assay, this is where our dendritic cell T-cell assays come into play. So just like in the MAPS assay, we generate monocyte-derived DCs, again from panels of healthy donors, typically 20 to 50 different donors. The donors are all typed. We load the dendritic cells as immature DCs with the protein of interest. We need a smaller amount of protein compared to the MAPS assay. For, I think, a 40-donor study, it's something like 10 milligrams of protein. They're all run at a fixed molar concentration in the assay. So depending on the molecular weight of the protein, it's also adjusted. And then the dendritic cells are matured, and then they're co-cultured with autologous PBMCs, so PBMCs from the same donors, which are then labeled with CFSE. And then we measure the proliferation by flow cytometry. And it's great, this assay, because you are taking into account this natural antigen processing and presentation. So although we're not determining what those peptides are that are presented, your whole protein is being processed in a natural way by cells from these healthy donors. And we've seen data, and there's more data coming out about the relevance of using healthy donors, these sorts of assays. It's always a question in the past, well, what's the benefit of that compared to patient cohorts? And certainly the studies that have come out to date, Bernard Mayer, Zubin was talking about it yesterday as well from their data, that actually when you do compare sort of disease versus healthy donors, the antigen presentation, the kind of the T-cell repertoire does tend to be relatively similar in both patients versus healthy donors as well. So what this assay allows you to do is to rank your proteins. At ProImmune, we generate what we call a response index, which is a combination of the percentage of donors responding, but also the strength of that response. So if you have a really potent T-cell response, but that only happens in maybe a very small number of donors, that's a little bit of a different answer to if you have a medium T-cell response in all of your donors. They mean different things. So that's something, that granularity is something that is reported and you can determine. And this is a study from a company called Elasmogen, which we're working with humanized shark VNAR domains. And they wanted to look at a number of their different lead candidates. And here we generate this response index. Typically, a lot of proteins will generate a response index from between about 0.1 and 1 in our hands. That in itself isn't really a problem. That's the general range. We do also get proteins which score 0. Occasionally we have something proteins that are completely blank with regards to T-cell activity. But generally speaking, most proteins tend to range in that sort of 0 to 1 kind of range. And then, of course, we have our positive controls for the assay as well to make sure that everything's working as it should. And I'm going to talk about cytokine storm. So ProStorm is an assay which we provide, which is really helpful for understanding when you've got particularly a final product or you've got a GLP batch of material. can this actually induce an unwanted cytokine storm? In the interest of time, I probably don't have much time to go through the introduction of the assay, but the main thing is that triggering of cytokine storm, again, can be many different factors can contribute to that, and first infusion reaction is really a key challenge. And the FDA are very hot on this, of course, and we see a lot of questions coming to us in relation to new products that are being developed, new classes of molecule, but also existing classes, where these sorts of questions are very, very important. And they say specifically in the guidance that data from animal studies should be supplemented by in vitro assessment of cellular activation, including proliferation and cytokine release in human whole blood or PBMCs, and that measurement of a cytokine panel should be as broad as possible and include IL-6, interferon gamma, and TNF-alpha. So what we do in our ProStorm assay is we work with fresh, undiluted whole blood. So the donors are recruited directly for the specific study. And one of the great things at ProImmune is that we can start these studies very, very quickly. So typically, from when everything is initiated, we can actually get the donors in within a week and start the work. So if you've got your test article, there's usually a high time pressure behind these sorts of data requirements. And we can do the recruitment of our donors. The consent is obviously taken. We're drawing up to 50 mils of blood for each donor. And then within a couple of hours, that blood is then taken straight up into the lab, incubated with the test articles at a range of conditions in an overnight assay. And then we measure the production of those cytokines. So it's really as close as you can get to a clinical trial without actually having to do anything in vivo. So the blood is just drawn, and then we do the assay. The controls that we run are PBS as a negative control and staph enterotoxin B as our positive control. But we typically also run a number of comparators. So Erbitux as a low response comparator, and Campath, alemtuzumab, as a high response comparator. And then after the overnight incubation, we isolate the plasma, and then we perform a multiplex characterization using our ProArray Ultra platform to measure a whole set of different cytokines. So this is how the data is presented. We have our drug concentration. 100 micrograms is a top concentration. I mean, that's pretty high. We can adjust this. We can also do more than 4 concentrations if you need to as well. That's all flexible. And then what we have along the x-axis is the measured cytokine concentration. So this is looking at IL-6, and each of the different diamonds is a different donor. And you can actually look across the scale. So this top diamond is the same donor as you go, for example, through different drug doses. And then we have a vertical threshold, which is set based on 95% of donors being negative for Erbitux. So it's really just an arbitrary threshold that we place, so you can kind of see how, as you increase the drug concentration, the responses change. So this is just zooming out a little bit, looking at all the IL-6 responses against a whole set of different test articles. Here's our high response comparator. Even at a very low concentration, you can see all the donors are responding. And then, for example, if you aggregate a protein, this is Remicade, this is aggregated Remicade, that can also induce higher levels of cytokine storm. IL-6, IL-8, interferon gamma are probably the main cytokines that you really want to focus on, but we do obviously a wider panel as well. And the nice thing is we can then, of course, benchmark your protein. It's quite nice to do comparators, do it alongside other molecules, maybe of a similar class, ones that have already been approved for use, for example. And then finally, I'm just going to talk about MutaMap, which is a really exciting new technology. We've mentioned about immunogenicity, about re-engineering. We've talked about what happens if you want to change an amino acid and the impact that can have, for example, in the Vitreptocog alpha case study. But one of the really big challenges for protein designers and immunologists coming together is, well, if we introduce an amino acid change, that might be great for reducing immunogenicity. We can test that with these assays. But probably the bigger problem is the fact that the drug won't work anymore. Often the immunogenicity is going to be in key functional element regions of the protein, like a CDR domain. And if we then introduce a point mutation at that particular position, there's going to be a big problem. So MutaMap is a really great high-throughput method of looking at the impact of point mutations from a perspective of function and actually binding affinity to the target. What it allows us to do is it explores the effect of substituting each and every amino acid at each position in a protein sequence, one by one, looking at all 19 possible substitutions. That's not an absolute requirement. We can limit the number of mutations that we look at, but essentially that's what it can do. So we take your parental protein, we perform site-directed mutagenesis, we then perform in vitro translation, so the protein is expressed as an SCFV construct. We then carry out a high-throughput solution equilibrium titration assay. And the beauty of this assay is that actually the higher affinity interactions, that even the more exquisitely sensitive it is. So this is not Biocore, it's not that kind of assay. It's actually really an immunoassay which is based on a solution equilibrium titration. We then are able to identify optimized protein variants. So following this mutation and expression strategy, we then do the analysis of the activity and affinity by this multi-point solution titration assay, and we can carry out between 500 and 2,000 mutations in just 8 to 12 weeks. So it's really, really quick. This is an example of taking Avastin, obviously anti-VEGF antibody. Lucentis is obviously a well-known derivative of Avastin with just a small number of 5 amino acid substitutions performed in that particular antibody. And when we look at the data here for the Avastin in the solid line versus Lucentis, you can actually just see the improved affinity of Lucentis using the MutaMap system. But the reason that I'm going to show you this is actually when you look at the detail of the MutaMap of Avastin, you can actually see the kind of the power of this technology. So this is the CDRH1 and H3 domain of Avastin. And what we have here is a heat map of the data. So when we introduce a point mutation, so this is the glycine here at this particular position, if you change it to alanine, the affinity is worsened, greater than 3-fold. So that's what happens here. If you have glycine to glycine, obviously nothing's changed and it's exactly the same affinity. And if you have a green, that means actually the affinity has improved, greater than 3-fold. So you can visually generate a very, very quick map of what the impact of all these different protein substitutions are. And what it's clear is that there are some particular residues which are actually quite permissive to change and others which are not. Those that are not, if you look at this glycine here, you can see that every other change from a glycine is not good, apart from keeping it as a glycine. So there are some positions you just can't mess with. There are others which actually are quite good and give you a lot of different options. So options are what it's all about. It means that you can actually look at the effect of introducing a point mutation in your biotherapeutic, not just thinking about immunogenicity, but obviously improving affinity, potentially changing PK/PD to benefit you, but also expression, changing biomanufacturing challenges or improving biomanufacturing challenges, that there are really a whole set of different attributes that this can give you benefits to. For example, if I just zoom in on one of these positions here, this serine at 105, and just show you the raw data from the point of view of the affinity, you can see that this is the change to a valine actually gives you this improved affinity over the wild type. But actually there's a whole set of different sort of permissive mutations which you might want to look at if that was a requirement for you. And there are some that you really wouldn't want to look at. This actually is the Lucentis mutation. So serine to threonine is what was introduced, but in fact having the valine actually give you an improved affinity. So you can look at that and you can see whether that would give you a benefit in regards to immunogenicity, manufacturing, expression, all those different attributes. So the key benefits of MutaMap are really understanding which point mutations are permissible or beneficial in your protein sequence, including the quantitative determination of the impact of affinity and activity. It allows you to kind of really cherry pick the best mutations. And that can be applied to deimmunization, really in a very time-effective way. So it means you don't have to do one assay, then another assay, and then another assay. You don't have time for that. You really want to sometimes put all these different assays together. It means that we can alter cross-reactivity. Maybe you have a molecule which is at the moment not suitable for doing some of the development work. You might want to make sure that it's appropriate for use in some of the animal models that you're using. That can also be used for that. Improving humanization, or maybe some of these other engineered features, and improving stability, manufacturability through these introduction of deliberate point mutations. Of course, the other thing then it does is provide you with a unique IP as well. Delivery time is really key, and this is something which nobody else is able to provide, so it's a very exciting technology. So that sort of summarizes what I was going to say. Just thank you for your time. Really, drug immunogenicity is a key thing. We're going to be hearing about it all today, so I don't need me to say anything more about it. But we have a very broad experience with a whole range of different types of molecules, whether that's classical monoclonals, whether it's bispecifics or multispecifics in different formats, looking at biosimilars, comparing them to innovator molecules, antibody drug conjugates, vaccines, peptide molecules, different scaffold proteins, orphan drugs, enzymes, the list goes on. These are all things that we have a lot of experience with, and it'd be great to have a chat with you if you have any questions. But yes, I'd be very happy to take any questions if you have now. Thank you. Sophie.
Hi, thank you, Jeremy. I have a question regarding the integration of all of this data that we can generate using the different assays. Are you working at all on this? Having a score that would represent for one candidate protein, for instance, that would represent the immunogenicity risk based on all of this
Just a kind of overall
assessment, something integrated?
Yeah, it's a really good question. We're not. The big challenge, of course, with all these assays is the fact that you're working with different donor cohorts each time. And although the assay reproducibility on the whole is pretty good, clearly an advantage and a disadvantage is that you are working with different cohorts of donors each time. And trying to get that sort of measurement of a kind of fixed immunogenicity risk score, I think the big challenge is that it might just be oversimplifying things. But we definitely understand that that is something that people want to have. So it's just kind of trying to get it right in the balance of things. But thank you for your point, because that's it's well made, and obviously with all the in silico tools that are available as well, and actually integrating a lot of those strategies as well, it makes sense that ultimately we could maybe go to that kind of approach. Thanks.
Hi.
Sorry,
Thanks,
yes.
Jeremy. I have a question. In all the assays that you described, do you ever see a dose response? Do you ever try to do a dose response? And
yeah,
it just what's confusing to me is what sets the working concentration for the MAPS assay, for example.
Yep.
Right.
Okay. So the working concentration, the mass spec assay is like for an antibody, it'd be about 50 micrograms per mil final concentration in the assay. So if we take that assay as an example, it's a reasonably high concentration of the protein, but part of the reason for that is it's an artificial system at the end of the day. We're obviously working with monocyte-derived DCs rather than plasmacytoid DCs. We're really trying to skew the system to be able to identify the peptides that are actually being presented. So we're able to we want to use a concentration of protein which is obviously going to enable you to do that. There are dose responses. If you do reduce the concentration of the protein in the assays, then of course the number of peptides that you will identify drops off. But that concentration is one that has been optimized.
In the MAPS assay as well?
Yeah.
So if you go down concentration, you see less peptides?
You will see less peptides, yeah. Obviously that will depend a little bit on the protein, but yes, you will. The other thing, for example, in the other assay, like the dendritic cell T-cell assay, we're using a fixed molar concentration. And again, that's something which has been... when we go down to like a 10-fold reduction of that, then yes, you will obviously have a lower T-cell response to that protein. So actually keeping it the same is just one way of being able to do the comparison across multiple donors. But yes, you can do a dose response. That's not something that is impossible by any means.
Right. So then the question is, which dose is more reflective of what's happening in
Yeah. Well, it's a comparative assay at the end of the day, and I think that's the thing. So even if you're doing the T-cell assays and the peptides there are run at 5 micromolar, it's an assay that, if you're doing anything in the lab, these are all the peptide concentrations that people are running assays at, and they have been sort of optimized for the performance of the assay. We're not trying to make it sort of mimic what's happening in vivo. I think that's not what the purpose of it is. It's really just a comparison of the different proteins, and the way that it performs at those sorts of concentrations kind of gives it that robustness as well. But yeah, it's a good question. I don't think there's a perfect answer. You can always do more, but of course it costs more to do more, so it's a matter of just picking the best.
Hi,
Priya,
Jeremy.
sorry.
Thank you. The Muta map, can it be used? So when you have a TCR therapeutic, you heard Amy Rosenberg talk about the Titan situation. Mm-hmm. So can we use Muta map to do two things? One is if we see in silico hits based on alanine scanning, see whether they are genuinely of concern and are they being presented in that HLA type. And the second thing is our T-cell TCR therapeutics are restricted to one HLA type, right? Would you be able to use Muta to see how could that sequence be changed to expand it to a different HLA type?
Yeah. Possibly yes. That's not how it's been developed, but possibly yes. One of the challenges, of course, with the TCR-MHC interaction is that the affinity is way worse than an antibody binding to its target. So that really is the challenge. It's more of a sort of a technical one that would work. But theoretically, the principle of it, in the way that that you need to express the proteins in high throughput to make it work, but potentially yes.
Because in the TCR situation, a low affinity, if you don't see it in vitro, doesn't mean it's not happening in vivo, correct? So could you mimic the in vivo situation by having all the co-receptors and everything in place so that you can amplify a low affinity signal in an in vitro situation?
Probably that would be a bit of a challenge, because this is really just looking at that one interaction between another.
This is purely binding.
It's purely binding.
Okay.
We're not talking about. It's not a cellular-based assay.
Okay.
So
yeah.
that would be another league.
Okay. Okay.
Yeah.
Thanks. Yeah.
Hi, Jeremy. Great overview. Just a question on your assays where you involve looking at T-cell activation, especially for the storm and other kind of things. When you do that characterization of the cytokines, do you also go a little deeper and look at the kinds of T-cells being activated, whether they're central memory or effector memory cells?
Yeah, good question. We have done that once for a particular client that wanted to know that, so we can do that.
Okay.
The nice thing about it is that the donors that we're drawing from a kind of a local population, it's something which we can get consent to do, and we can draw blood not just for the cytokine storm but also to do flow cytometry analysis. So yes is the answer. We can do that, and we have done that before.
Well, the flip side of the question is, when you see this kind of activation, are there certain particular cell types that seem to be consistently activated in response to the antigens being presented or to the particular?
I don't think that was something that we specifically. That wasn't the specific question that we were looking at. So I don't have an answer, but I imagine there'll certainly be some interesting points to look at there.
Okay.
Yeah.
Somewhat of an associated question about the donors. A lot of this work everywhere across the world is being done on healthy volunteers.
Yeah.
But we are increasingly seeing the same drugs being used for different indications, so totally different patient populations. And the information that we collect in the preclinical stages is usually on healthy volunteers. Do you see any or perceive any kind of shift to using more of disease subsets, PBMCs, or blood products?
I think not at the moment, certainly. It's for two reasons. I think we're also seeing, though, that the data is coming out where you are comparing disease cohorts to healthy donors. That actually, I mean, maybe it's what we want to see, but I do think that working with healthy donors is actually still a very justifiable and actually increasingly so approach. Because the challenge with using patient samples is predominantly, it's a combination of both logistics but also kind of ethics and everything else. So it really just becomes just not really a practical solution. And of course, there's no point in doing something just because that's the only way you can do it. But on the flip side, I think we are really seeing that actually that's generating very valuable data that is then backed up when you are looking at patient cohorts as well. So although we do have access to certain small numbers of patient type cohorts, it's never going to be at the same scale as we're able to achieve with the healthy donors. Shivani, yeah, thank you.
So when you report parameters like expect value or your response index, are these like generally accepted when you file reports with regulatory agencies, or this is something like unique?
Yeah. Okay, so there's a few points there. So the expect value is definitely is a standardized term in relation to proteomics. The response index is not. That's something which we as ProImmune have devised. But in answer to your question about the agency, they have seen data from all of these different assays, probably most of all the ProStorm assay, but also the DCT T-cell assay and the MAPS assay. and we've never had any comments back in relation to how to, I mean, obviously there are always questions of how to interpret data, but we've not had any specific questions back that that's an issue or a particular problem. Yeah, thank you.
Hi, Jeremy.
I have to make this the last one. I don't want to hog the floor as I get another speaker on. Yes, sorry.
I have a technical question regarding the T-cell proliferation.
Mm-hmm.
Did you do any depletion studies, like for example, like for you look for CD4 T cells? Did you deplete CD8 and then look for CD4? So do you see any difference with whole PBMC as such for CD4 T cell proliferation, and then compared with the CD8 depletion with the different HLA types or response index when you compare all these?
I don't think we've done that direct comparison. We've certainly, I mean, the CD8 depletion is carried out on the T cell proliferation assays in advance of obviously using the cells in the assay. But we also have done a lot of whole PBMC T cell proliferation assays, particularly when you're using shorter peptides, limers, for example, because obviously you're then focusing as well on the CD8 T cell responses anyway. We can certainly look at those questions, but that's not been a sort of direct question we've looked at. Yeah.
Thank you.
Maybe we can have a chat later as well. I'm going to have to move on just so that we carry on today. Thank you very much for your questions.